Recombinant DNA technology complete practical videos. Following is the list of the experiment performed. Let's begin with the practicals. Purification of plasmid DNA using alkaline lysis method. This is the principle of plasmid isolation and purification. We are going to use overnight bacterial culture. Pour the culture in vial. Centrifuge it for 5 minutes. Remove the supernatant from the tube. This is the composition of solution 1, 2 and 3. Add 150 ml of solution 1, that is resuspension buffer, in tube. Vortex it for few minutes. Add 200 ml of solution 2, that is lysis solution. Add 150 ml of solution 3, that is neutralization solution. Store the tube in ice for 5 minutes. Centrifuge for 10 minutes. Take the supernatant and transfer to the new microfuge tube. Add an equal volume of phenol and chloroform. Again vortex it. Centrifuge it for 2 minutes. Transfer the upper layer to the fresh tube. Add 2 volumes of ethanol at room temperature. Again vortex it. Centrifuge it for 2 minutes. Remove the supernatant completely. Add 1 ml of 70% ethanol. Centrifuge it for 2 to 3 minutes. Remove all of the supernatant by aspiration. Store the open tube at room temperature. Add 50 ml of nucleus-free water. Measure 12 ml of 50x TAE buffer solution. Measure 588 ml of distilled water. Add 12 ml of 50x TAE buffer solution. Weigh 1 gram of agarose powder. Dissolve agarose powder in TAE buffer solution. Heat it. Add ETBr, which having the concentration of 0.5 mg per ml. Cool it. Pour agarose in casting tray. Carefully place a comb in gel. Wait for solidified. Pour 50x TAE buffer solution in gel box. Add 4 ml of loading dye in sample. Carefully add 24 ml of loading sample into wells. And plasmid is isolated successfully. Restriction digestion of PUC19 using EcoRI restriction enzyme. In restriction digestion, restriction enzymes such as EcoRI binds DNA at a specific recognition site and cleave it and make it sticky ends. Let's start with procedure. Components we are going to use is PUC19 plasmid, EcoRI restriction enzyme, 10x SA buffer, 100x BSA and nucleus-free water. All these components were added in sequence shown in the given table. First of all, add 41.5 ml of nucleus-free water. Then add 5 ml of 10x SA buffer, which having the concentration of 1x. Add 0.5 ml of 100x BSA, which having the concentration of 1x. Add 2 ml of 270 mg per ml of PUC19 plasmid. Then add 1 ml of 20 units of EcoRI restriction enzyme. Centrifuge it for few seconds. Incubate the vials at 37 degrees Celsius for 2 hours. This is how sample looks after incubation. For analysis, the completion of digestion with the help of agarose gel electrophoresis. Now add 20 ml of sample into fresh tube. Add 4 ml of loading dye. Carefully add 24 ml of loading sample into wells. Run the gel at 80 to 150 volt for 1 hour. And here we have completed restriction digestion of PUC19. Digestion of PUC19 using BamHI and Ssp1 restriction enzymes. Listed plasmid are the plasmid that undergo double digestion with 2 different restriction enzymes. These ends are not compatible with each other and do not undergo self-ligation. After digestion of PUC19 plasmid, BamHI makes sticky ends and Ssp1 makes blunt end. So we are going to add this component in the sequence as shown in the given table. First of all, add 11.8 ml of nucleus-free water. Now add 2 ml of 10x SA buffer, which having the concentration of 1x. Add 0.2 ml of 100x BSA which having the concentration of 1x. Add 4 ml of 200 nanogram per ml of PUC19 plasmid. Add 1 ml of 10 units of BamHI enzyme. Add 1 ml of 10 units of Ssp1 enzyme. Place the tube in ice for 1 to 2 minutes. Centrifuge the vial for few seconds. Incubate the vial at 37 degrees Celsius for 1 hour 15 minutes. For analysis, the completion of digestion by agarose gel electrophoresis. Add 20 ml of sample into fresh tube. Add 4 ml of loading dye. Carefully add 24 ml of loading sample into wells. Run the gel at 80 to 150 volt for 1 hour. And here we had confirmed restriction digestion of PUC19 by BamHI and Ssp1 restriction enzyme. Purification and quantification of plasmid DNA. This diagram showing the interaction between the phosphate group of DNA, sodium ions, iodide ion and silica bit. Weigh 2 ml of vial. Extract the plasmid from gel. Weigh the vial after adding agar gel and plasmid DNA. Add 1.132 ml sodium iodide solution in vial. Keep the vial in water bath for 5 minutes. Add 15 ml of silica bits. Invert the vial for proper mixing. Centrifuge the vial for 30 seconds. Store the supernut in separate vial without disturbing palate. Add 200 ml of washing buffer. Vortex it for few seconds. Centrifuge for 30 seconds. Discard the supernut. One more time add 200 ml of washing buffer. Vortex it. Centrifuge for 30 seconds. Discard the supernut. Keep the vial in dry incubator for 10 minutes at 55 degrees Celsius. Add 20 ml of nucleus-free water. Resuspend the palate by mild vortexing. Incubate the vial for 3 minutes in dry incubator. Centrifuge for 1 minute. Collect the supernut in another fresh vial. To check whether the purification was done properly or not with the help of the UV spectrophotometer. 10 ml of purified DNA was diluted with 2990 ml of distilled water. Absorbance was recorded at 3 wavelengths, 230, 260 and 280 nanometer. And here purification of gel and quantification of the gel is done successfully. Blue-white screening. Here this diagram represents the PUC19, that is 2686 base pair plasmid map. This diagram showing the reaction between the X-gal and beta-galactose. It represents the construction of cation bridge and pore formation on the outer wall of bacterial cell. This diagram represents the functional and non-functional beta-galactose to give two different colored colonies, that is blue or white. Let's start with procedure. Weigh 20 mg of X-gal. Weigh 23.8 mg of IPTG. Add 1 ml of TMSO in X-gal. Add 1 ml of distilled water in IPTG. Vortex it. We are using LB agar in 100 ml of distilled water. Add 100 ml of ampicillin in broth. Then add 100 ml of IPTG. Add 100 ml of Xgal. Stir it well and pour the media into Petri plates. Culture were kept on ice for half an hour. The culture was divided into two tubes equally. Centrifuge the tube for 10 minutes at 4000 rpm and then 6000 rpm. The supernatant was discarded. The pellet was resuspended by adding 5 ml of ice cold 0.1 molar CaCl2 in each tube. Centrifuge at 6000 rpm for 10 minutes. Supernatant was discarded without disturbing the pellet. Resuspend the pellet with 0.1 molar CaCl2 and glycerol, adding 800 ml in each tube. Arrange the vials on ice. 100 ml of culture was collected into 1.5 ml vials. 10 ml of ligated sample and 10 ml of control added to separate 100 ml of competent cell. Tubes were kept on ice. Both the tubes were placed at 42 degrees Celsius for 90 seconds. Immediately kept the tubes on ice. 800 ml of LB broth was added to both tubes. The tubes were kept at 37 degrees Celsius for 35 minutes in a shaking incubator. Centrifuge it for few seconds. Randomly discard the supernatant and resuspend the pellet. 100 ml of transformed and control cell were separately plated on Xcal IPTG lureal agar plates containing the ampicillin. Incubate the plate at 37 degrees Celsius for overnight. Ligated material was successfully transferred in PUC-19. Very few blue colonies and a large number of white colonies were recovered. Molecular cloning of green fluorescent protein in EcoRI. Formation of the recombinant plasmid by ligation of the vector and GFP gene digested with EcoRI. First of all, we will prepare LB broth and agar broth. Measure 1.5 gram of agar powder. Add 1.5 gram of agar powder in 100 ml of distilled water. Measure 12.5 gram of LB for 500 ml of distilled water. Stir it well. Add 400 ml of LB to 6 gram of agar. Now add 100 ml of LB to 1.5 gram of agar. Autoclave it. Pour the media and allow it for the solidify. Now set up a reaction for ligation. Add all these components as shown in the given table. Add 11 ml of nucleus free water. Add 2 ml of vector which having the concentration of 3 nanogram. Add 4 ml of insert DNA which having the concentration of 7 nanogram. Add 2 ml of ligation buffer which having the concentration of 1x. Add 1 ml of T4 DNA ligase which having the concentration of 2 units. Vortex it for few seconds. The vials were kept in refrigerator at 4 degrees Celsius for overnight. Allow the bacterial culture to grow at 37 degrees Celsius in shaken incubator. Culture is transferred equally into two tubes. Centrifuge tube for 10 minutes. Carefully discard the supernatant completely. Add 1 ml of ice cold CaCl2 which having the concentration of 20 mm. After resuspension, add 8 ml of ice cold CaCl2. Place the tube on ice for 20 minutes. Centrifuge it for 20 minutes. Discard the supernatant. Again add 850 ml of CaCl2. Aliquate 100 ml of suspension into 8 tubes. Place it on water bath for 10 minutes at 65 degrees Celsius. 10 ml of ligated sample is added to the 100 ml of competent cell. 1 ml of control DNA is added to it and kept the vial to ice for 20 minutes. Kept the vials in water bath for 2 minutes. Immediately kept the vials on ice. 0.5 ml of LB broth is added to both vials. Keep the vials in shaking incubator for an hour at 37 degrees Celsius. 100 ml from the ligated vials was aspirated. Spread the ligated sample and control on respective LB and ampicillin plate. Plates were kept in incubator for 24 hours. Here's the result. GFP gene transformation produce green fluorescence on LB and ampicillin plate under UV light. All these experiments were done in DYPIU labs under the guidance of Dr. Priyatos Ranjan sir. Thank you for your valuable guidance and supporting us.